So I want to welcome everyone back. This is going to be our screencast for Chapter 23. And in Chapter 23, we're going to take everything that we have learned in Chapter 22, and we're going to apply that idea of evolution or change over a period of time. We're going to apply that idea to population. Now, typically when you think about evolution, you think about evolution occurring over a very long period of time. In other words, we're talking about maybe hundreds of thousands, if not millions of years. But in this case, we're going to look at evolution on a microevolution scale. In other words, we're going to look at a change in those allele frequencies. In other words, those little bits and pieces that actually code for the different traits you would find in an organism in a population over many generations. So we're not going to have to spend quite as much time to see that change occur. Now, in order for this evolution or change to occur, remember that we need to have something called variation within our population. Because remember, variation is going to be considered one of the raw materials necessary for evolution to happen. Now, there's going to be two main sources of variation that we'll look at. One of those will be chromosomal mutations. In other words, those mutations that are going to delete, disrupt, or rearrange those loci. The second is going to be sexual reproduction, because remember, sexual reproduction gives the species the opportunity to actually shuffle those existing genes or alleles into new combinations. Now, down here towards the bottom, this is just kind of a really quick example of the idea of microevolution. In other words, when you look at these two beetles, both of these beetles are green, but because there was some sort of mutation that occurred, we actually ended up with a new phenotype over here on the right. Because if you look, most of these beetles are green, but we have one here that's kind of green and actually has a new mutation that makes it a little bit brown. And so now we've introduced a different type of allele within that population. So what we would do is we would look at the frequency of that allele over many, many generations to see exactly what that particular allele might do to that population. Now, sometimes what we can do with populations is we can actually apply a little bit of math to it to sort of determine how fast or even if evolution is actually occurring within that population. Now, for a person that actually studies this type of information, they're going to enter an area of biology called population genetics. So population genetics is going to be the study of genetic variation among populations. Now, we need to have a good idea as to what a population is. Now, a population is going to be a localized group of individuals, and again, it's going to be a species in a particular area that is going to be capable of interbreeding and producing fertile offspring. The other thing we need to look at is something called a gene pool. Now, a gene pool is going to be basically all of the alleles for that particular loci or gene in a population. Now, we need to think about that locus or loci carefully because sometimes those locus or loci will be fixed. And what we mean by that is if you think about a particular gene or a genotype for a particular characteristic or trait, if the genotype would happen to be homozygous dominant, all individuals were homozygous dominant, then it would make sense to you that the particular locus is going to be fixed for that particular characteristic. Unless, of course, we have a mutation that occurs within that particular loci. Same thing could occur for two lowercase letters. In other words, we could have homozygous recessive, and so in that case, it could be fixed. In other words, unless we have some sort of mutation occur. Now, something else we need to think about here is something called the frequency of an allele in a population. And again, that's just looking at the likelihood that you would actually find that allele in that population. And as I had said, we're going to do a little bit of math to calculate that particular frequency. Now, of course, if there are two alleles at a particular locus, what we're going to have to do when we do the math is identify them by a particular identifier or letter. And in this case, the P and the Q are going to be used to represent their frequencies. Now, if you look on this screen, you're going to notice we do have our Hardy-Weinberg equation that is going to be represented, and it's going to be right here. Looks a little bit intimidating, but please don't let it intimidate you because we're going to work through a problem on the next screen that kind of exemplifies or shows you what we do with this particular equation. Now, sometimes this Hardy-Weinberg equation will be described as the Hardy-Weinberg Principle. Now, the Hardy-Weinberg Principle simply is describing an ideal population. So that would be one that is not evolving. And so what we're going to do is we're going to compare the numbers that we get from this equation to what we would consider the ideal population. Now, the closer a population is to the criteria of the Hardy-Weinberg Principle, the more stable the population is likely to be. Now, these criteria that I just mentioned are right down here towards the bottom. So the 5 criteria for Hardy-Weinberg is going to be basically there can be no mutations occurring within the population, random mating must be occurring, no natural selection can be happening within that population. In other words, every individual within that population has an equal chance of surviving. There needs to be an extremely large population to sample from, and there can be no gene flow. In other words, no immigration or immigration in that population. Now going back up here towards the middle, you're going to notice we have each of our letters being identified over here on the left. So when you look at the P within the equation, that's going to be the frequency of the dominant allele. The Q is going to represent the recessive. When you look at P squared, that's going to be the frequency of the individual that is the genotype homozygous dominant. Q squared is going to represent homozygous recessive. And in this case, when you look at 2PQ, that's going to be the frequency within that population of the heterozygous individual. Now, something to be aware of is that the frequency of all alleles in a population are always going to add up to 1. So P plus Q is always going to equal 1, and that's going to be important when we look at the next screen. So the easiest way for us to understand the Hardy-Weinberg principle is actually to go through a sample problem. Now what we're going to do here is we're going to use the one that was in your textbook, and this is the one that basically looks at the condition called PKU, or phenylketonuria. Now this is actually a condition that is tested for when every baby within the United States is born. And this is a condition where the person actually has some difficulty breaking down the amino acid called phenylalanine. And because of that difficulty, they have a very restricted diet. Now what we need to do is we need to look at the 5 criteria for Hardy-Weinberg equilibrium. So in other words, this would be the ideal condition where evolution would not be occurring. So in this case, and for PKU, we need to make the assumption that the PKU gene mutation rate is going to be very low, because remember, no mutations can be occurring. Mate selection needs to be random with respect to whether or not an individual is a carrier for the PKU allele. In other words, we can't have individuals actually reproducing that know for a fact that they're going to produce children with PKU. Number 3 is natural selection can only act on rare homozygous individuals who do not follow dietary restrictions. In other words, most people, if they are diagnosed with PKU, will be following their strict diet. Number 4, the population needs to be large. And number 5, migration has no effect, as many other populations have very similar frequencies for this particular condition. Now, if you look at the example, what we know is that PKU itself occurs in about 1 in every 10,000 births. So what we do with that number is that we simply take 1 divided by 10,000. That's going to give us this number that you see right here. And so this is going to represent the individual who has PKU. So in other words, Q squared is going to be the homozygous recessive. What we want to do is we want to look at the frequency of that particular allele within our population. So we simply take the square root of Q squared, in other words, of this number, and that's going to give us 0.01. So that's going to be the frequency of that allele within our population. Now, of course, now that we know the frequency of the recessive allele, we could easily determine the frequency of the dominant allele. In other words, that P designation that you see right here. Now remember, P plus Q is going to equal 1. So what we do is we simply do a little bit of rearranging. P is going to equal 1 minus Q, so 1 minus the 0.01 that we had up here. So that tells us that the frequency of the PKU allele is going to be 99% basically, in other words 0.99. So the frequency of the heterozygote carrier is going to be, again, the 2PQ. So we simply take 2 times the 0.99 times the 0.01. So in other words, each of these were our alleles that we had looked at. And so the frequency of the heterozygous condition, in other words, capital A, lowercase a, is going to be 0.0198, or approximately 2% of the U.S. population. Now, there's going to be 3 major factors that actually can alter those allele frequencies that we looked at in the previous screen. And the 3 major factors that are going to alter these are going to bring about the most evolutionary change within a population. One of those, of course, is going to be natural selection, and they consider this a non-random type of factor. So remember that natural selection represents differential success in reproduction. In other words, only those that have the ability or opportunity to reproduce are going to contribute their alleles to the population. Then the second is going to be something called genetic drift. Now this is considered a random type of situation, and genetic drift simply describes how allele frequencies are going to fluctuate randomly from one generation to the next. Now, the thing about genetic drift is that the smaller the sample, there is a much greater chance of deviation from what we would consider a predicted result. Now, genetic drift also tends to reduce genetic variation through the losses of some alleles within that population. Now, the third thing we need to look at is something called gene flow, and this is going to consist of the movement of those alleles among the populations. Now, this diagram that you see right here actually is a good representation, and again, this one was just taken from your textbook, of genetic drift. If you notice, we're looking at the frequencies of the dominant and the recessive, so we have 0.7 or 70%, 0.3 or 30% of these alleles. And if you notice, in this population right here, the ones that are actually boxed in, these are the ones that actually had the opportunity to reproduce. In other words, they were able to produce fertile offspring. The other ones that you see here were not able to do that. So if you move into the next generation, what you're going to find is the same thing happens. But if you notice, our frequencies for these two alleles changed a little bit. So now we're at about 50-50. But again, these individuals did not have an opportunity to reproduce, but as I had said in the previous example here, the ones that are boxed in actually did produce fertile offspring. And because they were the only ones that were able to contribute to the next generation, that definitely changed the frequency of the P and the Q, where now all we have is the dominant allele, and basically the recessive allele has left the population. So this would be a really good example of genetic drift. Now, when you look at genetic drift, what we do is we actually have two categories for that. We have one called the founder effect, the other called the bottleneck effect. Now, the founder effect is going to occur when a few individuals become isolated from that larger population. So if you look over here, you can see a circle within this larger circle. And if you notice, if we isolate those individuals, what you see here is very different from what you see on the mother population on the left. So when these individuals begin to reproduce, it's going to make sense, hopefully, that we're going to get a lot more individuals that are going to be the yellowish orange dots as opposed to the brown dots that you see here. So the allele frequencies in the small founder population can be very different from those that were in the larger parent population. Now, the second example of genetic drift is going to be something called the bottleneck effect. And this is where you have a sudden reduction in population size due to the change in the environment. And oftentimes this is going to be caused by a natural disaster. So the resulting gene pool may no longer be reflective of the original population's gene pool. And if the population does tend to remain small, it could drift even further. So over here on the right, again, this is an example taken from your book. This is the original population. So if you look at all these different colored marbles that are within this bottle, if we go to basically pour out these marbles, because the neck of the bottle is so small, that kind of restricts the flow a bit. And so what we have is we have something called a bottlenecking effect. So only a few of the marbles initially can actually come out of the bottle. Well, if you notice, the only marbles we actually had come out were the blue marbles and maybe one white marble. And so the surviving population, because of that bottleneck effect, actually is more blue as opposed to the mixture of the whites and the yellows and the blues. And so that's definitely going to affect the allele frequencies within that surviving population. So the effects of genetic drift, again, this is just a summary of what we might see, where genetic drift is going to be significant in very small populations. In other words, it's going to be more likely to happen. Genetic drift is going to cause allele frequencies to basically change at random. Genetic drift can lead to a loss of genetic variation within populations, and so in other words, we could see a pretty big change. Genetic drift can cause harmful alleles, unfortunately, to become fixed within that population. Now, we also have to talk about, as we had said, something called gene flow. Now, gene flow is going to basically center around something called immigration and emigration. So immigration is going to be the movement of a particular individual into a population, and emigration is going to be moving out of the population. So as we had said, gene flow consists of the movement of alleles among populations. Alleles can be transferred through the movement of fertile individuals, or they could actually be transferred by gametes as well. And a good example of this would be the movement of pollen in plants between different populations. Now, gene flow tends to reduce the differences between the populations over a long period of time, and it's simply because we have individuals moving back and forth between those populations. Now, gene flow is actually more likely than mutations to alter the allele frequencies directly within that population. Now, down here towards the bottom, what you see is we have two petri dishes One of them contains the beetles that are brown, the other contains the beetles that are a little bit lighter in color. Now, if you look here, you can see that these beetles can actually migrate between these different populations. So in other words, we're simply changing the allele frequencies based on whether or not an individual comes into the population, contributes to that genetic material, or exits that population, and maybe actually removes that genetic material from that existing population. So again, immigration and immigration is going to definitely affect the gene flow between those two populations. Now again, natural selection is the only mechanism that's going to consistently cause something called adaptive evolution. Now remember, this adaptive evolution is going to act on the organism's phenotype. In other words, what we see on the outside. It could be the way they look, or it could be the way that they behave. Now, the natural selection phrases struggle for existence, which is a phrase that we often use, and survival of the fittest, oftentimes can be misleading because what they do is they sort of imply to us that when you talk about natural selection, you're talking always about competition, and that's not necessarily the case. What we need to think about is we need to think about something called reproductive success. And sometimes this reproductive success can be very subtle, and remember that success can depend on many factors. Sometimes what we'll do is we'll use a phrase called relative fitness, and this is going to be the contribution an individual actually makes to the gene pool of the next generation relative to the contributions of other individuals. And so in other words, you would see a difference between the fitness of those individuals within that population. So selection is going to favor certain genotypes, and it's going to favor those genotypes by acting on the phenotypes of other organisms within that population. Now, when you talk about natural selection, there's actually three different modes of natural selection. We have one called directional, one called disruptive, one called stabilizing. When you look at directional selection, this is going to favor individuals at one end of the phenotypic range. So down here towards the bottom, this is going to be what we would consider the original populations. So we have 5 different phenotypes in regards to the fur color of the mice that you see here. Now, when you see the distribution of this population, what you're going to see is you're going to see instances of each of these different color varieties. But when you have something called directional selection, you're going to find that sometimes what we'll do is we'll sort of favor one end of the spectrum. In other words, one end of that phenotypic range. So in this case, we're favoring the darker colored mice as opposed to the lighter colored mice. Over here, when you talk about disruptive, what we do is we tend to favor both extremes, but maybe we don't favor the intermediate colored mouse that you would see up here. Then if you notice over here we have something called stabilizing, where the intermediate variants are actually favored while the extremes are being selected against in that particular population. And again, it's all based on the environment that these organisms happen to live in. Now we need to remember that natural selection is going to increase the frequencies of those alleles that are going to enhance survival and reproduction. So adaptive evolution is going to equal the match between an organism and its environment. So we need to remember that environments are always in a constant flux or change, so adaptive evolution is going to be a continuous process. Genetic drift and gene flow are considered random, so they do not consistently lead to adaptive evolution, as they can increase or decrease the match between organisms and their environment. Now over here on the right, you're going to see a couple of examples of adaptive evolution. One of those is the color changing ability in the cuttlefish, and this is going to be a type of a mollusk that you would actually find in the ocean. And what they do is they actually have the ability, based on the cells that they have in their skin, to change the color or texture of their skin. And when they do that, they're able to blend in really well with the environment that they happen to be in. And so that's a type of adaptive evolution. They adapt to their environment. Over here, if you look, you have the movable jawbones in the snake. It basically gives them the opportunity to swallow really large prey. Now, sexual selection is also something that's really important to look at because it has a very heavy influence in natural selection. Because if you think about it, you're basically trying to achieve mating success. Now, what this does in the animal kingdom, it results in something called sexual dimorphism. Now, sexual dimorphism is simply very marked differences between the sexes in the secondary sexual characteristics. And that's going to be something that's very obvious when you look over here at the peacock that you see in this picture. The females are typically very drab in color, but the males definitely have a very vibrant and very extensive type of display. And based on that display, the female gets to choose whichever one she thinks is best. Now, of course, that's going to influence the genes that are going to be passed on to the next generation. So again, you got to think about this because male showiness, again, is going to be something that's really important to attract that female, but unfortunately, to be that showy could possibly also decrease his chance of survival. So in other words, you have to think to yourself, so how do female preferences actually evolve? Well, what they have is they have something called the good gene hypothesis. And what this is going to suggest is that if a trait is related to male health, both the male trait and the female preference for that trait should be selected for in that population. So in other words, both of these traits actually work in tandem with each other to remain in the population. Now, sometimes we need to think about these genes as actually remaining in a population. So what they refer to is something called the preservation of genetic variation, because sometimes it actually becomes beneficial to have what we would consider a harmful allele in our population. So there are various mechanisms to help preserve genetic variation in a population. One of those is called diploidy, and you often find this in plants. And this is going to maintain genetic variation in the form of what we would consider recessive alleles within that population. Now, something else you can consider here is something called heterozygote advantage. Now, this is going to occur when heterozygotes actually have a higher fitness than do both homozygotes. So natural selection will tend to maintain two or more alleles at that particular locus or site on that particular chromosome. So the sickle cell allele is something that's a really good example of a heterozygote advantage, because anybody who has this particular allele actually has some protection against something called malaria. So they have malaria resistance. So if you look over here on the right, this is going to be a good example of heterozygote advantage. Now, the biggest thing to take away from this particular diagram is that if you notice, anywhere that you actually have a pretty large or pretty high incidence of that malaria-causing parasite, you also tend to find people that have the heterozygote genotype for sickle cell. And again, it's because for whatever reason, it tends to provide those people some protection from getting the malaria that would be caused by that particular parasite. Now, another thing to think about when you're talking about natural selection is something called frequency-dependent selection. So in frequency-dependent selection, what we have is we have the fitness of the phenotype, but it's going to decline if it becomes too common in the population. So in other words, selection is going to favor the phenotype that is less common in a population. So what we have here is we actually have a couple of examples of fish. These are considered right mouth. These are considered left mouth fish. Now, the reason that becomes important is because when you think about the right-mouthed fish, what they do is they attack their prey. Sometimes what they're called the scale-eating fish. And what they do is they attack their prey actually from the left-hand side. But what happens is when you have a prevalence of these right-mouthed fish and you have prey that are being attacked from the left-hand side, those prey tend to learn. And what they do is they start to sort of guard their left-hand side. And so because they start doing that, they're protecting themselves from this particular variety of this fish. And so over a period of time, what happens is these fish start to not be quite as common, and the left-mouthed fish, which are still there, actually start to increase in numbers. But then over a period of time, once these fish start to feed, they actually attack the right-hand side of the fish. The prey fish, actually start to look at the right side and protect their right side. So basically what they do is they escape, and so in other words, these fish start to decrease in numbers, and the ones that are still present, the right-mouthed fish, now these start to actually increase in numbers. And so what you see is you basically see an up and down in regards to the frequency of the numbers of these types of fish over a long period of time. So the last one we're going to look at is something called neutral variation. And neutral variation is simply genetic variation that appears to have no selective advantage or disadvantage in the population. And fingerprints are a really good example of this. There's a lot of different varieties of fingerprints in our population, but again, it doesn't really cause you any type of disadvantage to have this type of fingerprint over this type of fingerprint. And also, it really doesn't give you any type of advantage to have this fingerprint over this type of fingerprint. So they would consider that a neutral type of variation within a population. All right, so we need to remember that natural selection and everything that we've talked about up to this point is never going to be able to fashion what we would consider a perfect organism. Selection is only going to act on those existing variations within that population. Evolution is going to be limited by those historical constraints. In other words, we need to think about descent with modification. So in other words, it can only work with what is there. Adaptations are often compromises. And again, chance, natural selection, and the environment are all going to interact together to encourage that change to happen. All right, so as always, please make sure that you have completed your screencast study guide.